Hello BIO178 and welcome to another lab module. So in lab this week, what we will be doing is continuing to work with the samples that we have been for our group project. So last week what we did is PCR to amplify that 16S region of the different bacteria that were present in our samples. And what we're going to do this week is run those PCR samples out on an agarose gel. By running it out on a gel, we'll be able to see if there's a band that shows up. Basically what we're doing in this step is checking to see if a band shows up. If it does, that indicates that we have sufficient 16S DNA in our samples to then send out for sequencing. So we'll be running out those 16S samples on a gel, looking at those gels to figure out which of our samples have a strong band and therefore are sufficient to send out for sequencing. And then we will gather all of our DNA in one place that we agree is good to send out. And following this, we will send out the samples to some of my collaborators and they will then do high throughput sequencing of these samples and we'll get some really interesting data back for us to analyze. So per usual, go ahead and click through this module, take a look at the written lab instructions, and also take a look at the lab report template so you know what information you need to make sure to note down and record for your lab report this week. And like always, if you have any questions, let me know. Otherwise, I look forward to seeing you all in lab, and I'll see you soon.